than 10 years now. And the vision of this whole project is to manage very complex scientific research. Here's an example of a complex reaction network, a complex reaction synthesis route of a pharmaceutical. And if you want to understand all these details, for example, all the connections between these reactions, the kinetics of these reactions, and if you even want to replace such reactions by more biological-based reactions like enzymatic reactions, like with an enzyme shown here, so transaminase, which was done, then you need to understand a lot about this reaction. You need to understand all the components, you need to understand the kinetics, the reaction mechanisms, and you also need to understand the biology or molecular biology to make these enzymes. So there's a lot to understand. Or another very complex thing is our metabolism. So if you want to understand all these connections between the enzymes that are involved in such a very complex network of reactions which happens in every second in our body, then you need also a huge knowledge base and a huge experimentation that can store all this interlinked information. Sorry? All this connected information. And here's something completely different. Imagine that you want to study the development and the history of mansions, so of old castles in Germany, for example, and want to reconstruct these, then you need a database and a place where you can collect such information in one place. And this is also part of our projects that we're doing. So it's a completely different thing. So we're doing also 3D architecture reconstruction with Gaussian splatting. So what these things have all in common is that the data we are producing and which we are looking at and where we are learning from is very highly interconnected. You can see that these experiments are connected to the devices that do these experiments, to the substances, to the organisms. The experiments are related to projects. Projects have sub-projects or experiments. So all of that, all these interconnections need to somehow represented in a data management system. And since such a system didn't exist when I started and still doesn't exist, I started to develop LARA. And so how does such a thing look like? I mean, from an artistic perspective, modern comic, you could think like an assistant that helps scientists to do the experiments. But on a more technical perspective, this whole infrastructure looks like this. It starts as every scientific experiment, as every research with planning of projects. Then you execute somehow your experiments, and we are utilizing the CELA infrastructure, which is which stands for standardization in lab automation, and is used to communicate between lab devices, but not only with lab devices. It can also communicate with humans. It can also communicate with machine learning algorithms, with frameworks that do machine learning. Then these data orchestration, once it's running, the data that is produced is collected and stored in databases. And we have two representations: a classical relational database. So in our case, it's PostgreSQL database. And this mainly contains information that is tabular, like bulk mass information about, for example, molecules, so chemical information or sequences. And then we have semantic representation, which mainly contains metadata about the experiments in triple stores, so in a graph database. And this enables then SparkQL queries to these graph databases. All of that is now then connected to with MCP protocol, so Model Context Protocol, which is a quite recent development, as a standardized interface to large language models. In our case, it's Qwen. So we are running this Qwen locally on our university. And then the ability, the interfacing of the database with the machine learning algorithms can then be done through the MCP protocol and through the application of the large language models. And since everything is open source, open standard communication, open metadata standards, you can build a generic closed loop system. That means that the AI can advise new experiments, can plan them again, and ask the orchestrator to execute these in an agentic way, these workflows, and explore basically new scientific questions. Once the artificial intelligence develops in a way that you can really build hypotheses, and they are at the brink of starting to do that, such an infrastructure can just out of the box do this closed loop experimentation and hypothesis building, so automated. Everything that you see here is open source. It's found on GitLab. So I'll come to this later. So now for a little demo, I recorded just to be on the safe side so it's a glimpse into our system, how it looks like. And what you see in the first place, so this is the login screen. I tried to build and include as many as possible NFDI services into the system because they are very useful, and it's also a showcase to see how NFDI services can be used. First service that you experience is the authentication and authorization service, which can be used to log in into LARA, so as authentication, similar to Google authentication or ORCID authentication. And once you log in, then you come to dashboard. And now I show you how this all looks like. So I now log into the system. I'm using the standard account now, the admin account, just to show everything that is available. What you see is a very plain dashboard where you can see the latest projects and you can click on them. You can explore them immediately. You can see the experiments related to that. You can explore the projects tree. So it's a whole hierarchy of projects that you build. And then you have tools. So one tool is, for example, Zotero. And what you see here is not your classical Zotero client that you have with which you install, but it's a front end which points to a self-developed Zotero backend. So it's a Dockerized version of Zotero. And this can be used then, this Zotero, to plan your projects. And then this is the panel of all these things that you can store in the database. So like the material, the organisms, the processes, the substances, the sequences, the samples. So whatever you want to store in this database can be stored. It's not like frozen, so everything is modular, so you can put modules in. And once it's in the database. You can operate with this data through a chat client. And I just put a prompt here, so I copy a prompt into the chat. So this is our chat system. We are using Open Web UI for that in the backend, and then you can interact with this LARA database through chat, so through LLM. So I start, for example, to plan a project by asking what I want to do. So in this case, I want to give advice for a PCR reaction. So and how this can be done. And when I advise the system, it will move this procedure into the LARA system. So since it's connected, so it's not like in your browser when you talk to ChatGPT. In this case, the information that the system is producing is interacting with it. And then it produces a workflow and an execution workflow and deploys it, in our case, to an execution system. This is Prefect. It's an open source Python-based execution system. And now you see how this workflow, this preparation of the PCR reaction is orchestrated. And it can be automated, so it can run then on a robotic platform, but it also can run by humans, so it can just send instructions. And you see here all the steps that are executed. So this is now the ready execution. So this orchestrator is a very nice tool, a generic tool to execute workflows and monitors them, Prefect executor. While this whole process running, we have like monitoring of the data, so online produced data. So what you see here is, for example, temperature data, and I added another trace of a light intensity data for a light sensor. And we have, since everything is generic, I mentioned that we are using Sila. This is the Sila open source Sila browser from Unite Labs. Since everything is open, you can just use standardized software to communicate with our devices. So this browser has not seen our thermometer, but still can get the current temperature, since it's an open standard and it's an open protocol where the Sila services explain themselves. So they explain what they are doing. They also send and this now this is now the data stored in this Sila in the LARA database. So these are all the snapshots that are produced during a run. And while this whole execution happens, you can sit in your lab. And this is just a mock-up, basically. Sit in the lab and monitor your the workflow. So you have you can can connect to a webcam, and then you look live streamed to the device that is currently executing the workflow. So in this case, it's a colony picker, our colony picker that picks colonies, but it can also be very complex devices. And if you don't want to see it anymore, then you can just click it away. And this is our robotic platform that we are monitoring. So I could look at the robot while it's executing from LARA, within LARA, as a kind of preview or no monitoring window. And I can build up multiple of these monitoring panels in the dashboard that I have, for example, a view on different things in the lab that are executed. If you have like an automation platform like ours, of course, this sounds futuristic, but more and more of these platforms are built, and they will be appearing in the lab. And if you look at large companies like Novo Nordisk or Roche or Novartis or Boehringer, they all have such platforms. So, and it's a very common thing, or will become a common thing. Yeah. So let's see. I think now it ended. Yes. Okay. So how is that now all connected? So I mentioned already that LARA is communicating with the large language models through the Open Model Context Protocol, MCP, and we're using a chat interface, and this can be either text prompts, or we can also talk with it through audio if you want, so that we can just in natural language communicate with the system. And then on top, we can add modules. In this case, a colleague of mine from Technical University of Berlin, Stefan Born, added, for example, a protein design and prediction platform, machine learning based. So it basically interacts with a large language model and then builds up the models for the protein engineering and instructs the system to collect the right data. And since everything is semantically annotated, this machine learning tools can extract the data directly from the databases, because everything is, as I said, interlinked and annotated. LARA itself has, as I mentioned a little bit before, a hybrid structure. So we have, for the data storage, we have bulk data storage, we have the relational database, and for the semantic representation, we have the ontologies. Everything is filled through these automated processes, and then large language models like our local large language model can communicate with these data through interfaces. Since we would like to query this data with only a unified system, like a SPARQL query, for example, we build up a hybrid architecture, query architecture. So we represent, for example, abstract knowledge like relations between entities in your lab, in your kind of research, through an ontology, and then the actual data is stored. Most of the actual data, those instance data, is stored in relational database, and we build up currently a framework that accesses the relational database through SPARQL queries. So we build up a system for SPARQL rewrites, basically. Very important in this whole organization is CELA. So CELA is like an USB. Imagine USB for labs. So it's basically you connect two devices. They announce each other. They say what they are, what they can do, and on top you can add features that expose the data that is produced in a semantically annotated format. So we are using for as a serialization format, we use a JSON LD as an RDF serialization. So the data that is produced, so every thermometer, every whatever measurement, every spectrometer sends its data already semantically enriched with JSON LD metadata, or in the data itself. Okay, and then another pillar in this whole FAIR data infrastructure is the persistent identifiers. For our system, LARA system, we are utilizing the PID for CAD, which is developed by David Linke from LICAD and Preston Rodriguez from HRIS. And this is a persistent identifier service that offers infinite amounts of DOI-like handles. So you know probably DOIs. So it's a similar system. And since this is free of charge, the usage of that, we prefer using that over DOIs. And you can identify all your data on a very fine-grained level. So so every sample can be it can have a PID. Every device, every substance, if you want, can have a PID. So it's very, very cost-free. The metadata itself, so the higher level metadata, we annotate with the DCAT AP derived profile, which is developed by Philipp Strömert and Henrik Borgel, David Linke, a little bit also myself. And it's for the whole, so it's a development that we do for the whole community, NFDI community. It's built on DCAT application profiles, and we basically do specifications of these application profiles for particular applications. So, for example, for chemical reactions, for cultural objects, for biological for animals or plant samples or whatever where you can catalog something, you can use this DCAT AP derived metadata scheme. And now comes something that it's not a lot, it's not mentioned a lot in the research community yet, but it's a part of LARA that is very crucial and very important. And this is blockchaining of data. So it's a little bit complex to understand, but once you grasp the potential of that, you see what this can change, how this changes in the long run science. So every data is basically hashed. So it's basically, so a checksum is a unique checksum is calculated over a certain amount of data. And this checksum is included in the next data block. I mean, how big this block is is defined by the developers. So that's arbitrary. But the important thing is once this unique identifier hash that is stored from previous block in the next one, you can't change this previous block anymore because it will change this checksum. And by that, once you record all these chains of checksums, there's no way to alter any more data. So this means, so data can be not only the data that is measured, but also ideas, texts that you wrote. So if you write a document and you note a very cool idea and you put it into a data block and it's hashed, then it's uniquely identified by this, and nobody in the world can change it anymore, except he has to change the whole chain. And you can uniquely prove that this idea originated from you and not from anyone else. So that's or this data was produced by yourself and not by any other lab. So it's a very very powerful method to uniquely identify your data and your ideas. Then once the data is stored in the LARA databases, we can apply the same mechanisms, these prefect workflows to evaluate the data. So we can do like simple statistics, or we can do machine learning with it. So whatever you imagine, all these workflows are, by the way, stored in Git repositories and can be versioned and can be called by the prefect engine on demand or in an automated fashion. So in data analysis pipelines. For example, if data comes from a automation system, we can advise the data analysis pipeline to trigger and to do some visualizations of the data. If you want evaluation of your individual data, of a particular one, not bulk evaluations, you can use the inbuilt Jupyter Lite notebooks. And this Jupyter Lite system is a new WebAssembly-based system to have like a complete Jupyter environment in your browser locally running. So it's not demanding any compute power from a remote server like the Jupyter hubs that are currently deployed all over the place. And and then once the data is then available, you can advise the machine learning models and AI to learn from, derive, and improve. The data itself is organized in knowledge graphs, as I tried to explain, and these knowledge graphs are built automatically while the data is coming in. So you build, and while you're organizing your research, while you're building hypotheses, these knowledge graphs are improved and built, and then can be queried. The last thing that you want to do with your data is, once you have like a good state of your data, you're happy with the results and you want to share it. We have plugins that then connect to various infrastructures like the Dataverse, or in development is also CCAN synchronization, or EOSC synchronization, or Zenodo. So with a click of a button, you can say then, of course, user selected, what data should be automatically synchronized to these remote repositories, and which metadata should be sent to them. So that means that once you have this infrastructure, you don't need to require anymore to enter all these metadata for these remote repositories anymore, because they are available from the system and they are sent automatically through the system. One example is our repo for CUT, so the repository from the NFDI for CUT, where we have like a Dataverse where we can put data on to share data. Yeah, and here's another repository. This is a GitLab repository of LARA. So if you are interested in the whole software development of LARA, it's all done open source in a modular fashion. So all of these modules that you see here represent some kind of knowledge that is stored in LARA, and they can be developed quite independently. Yeah, to sum up, I hope that I could show you how an AI-guided general purpose infrastructure could look like. I mean, of course, this is just one implementation, but it covers already most of these things that I explained, and it's a minimum set of functionality that is required to do these closed loop scientific explorations. The very important thing is that it should help you to focus on your science and not on metadata and research data management. It should, and it does that by having in the background all the semantics, all the knowledge, all the meaning of what this research is about, and instructs then helpers like agents, AI assistants, to do something with this data. And the very key of this whole infrastructure is that we build up fair data from the very beginning. So when data is produced from the devices, they speak SELA with the semantic information already, and it sends already it in automatically to the triple stores and build up knowledge graphs without interference of humans. So how can this evolve? One very important aspect I have in mind is, of course, which I repeated a lot, is this closed loop experimentation. And since this infrastructure is built to do that, I think in a very nearby future, we will be able to show how this all works. The blockchain technology that I mentioned deliberates scientists, democratizes data and ideas. That means that once you have like an idea and you put it into a blockchain data set, then you can uniquely prove it's your idea. It's not the idea of any of someone who tries to take it or something. So you can uniquely prove it. And this whole infrastructure with the data, this interlinked data representation opens a completely new way of publication. So going away from this PDF and Word and Excel-based publications to something decentralized, to something data-driven, to something where reinterpretation of the data is possible, because what we know and how we explain data is just our current perspective. But think in the future when more knowledge appears, data could be reinterpreted. And since everything is then available and structured, you can apply technologies like modern AI technologies, reasoning technologies, to explore legacy data in a completely different perspective. And this is, I think, a very powerful progress that can be achieved with such a system. To close, I have to thank a lot of people. Of course, you can imagine that a lot of this can't be done by one person. This develop. So a lot of people from so, for example, the SELA people or the people from our IT department developing this large language models and compute infrastructure, the HPC. Then, of course, I couldn't develop so quickly if I hadn't such tools like Claude or Gemini or Copilot. And then the knowledge communication, knowledge graph building and semantics is a very complex thing, ontology formulation. So we are discussing a lot. So, for example, Henrik, David, and Philip, then the NFDI for CAD and NFDI for Chem helped me to inspire, challenge what I'm doing, and we try to include it. And NFDI for Base helps to provide services that I don't need to provide anymore. And finally, also the European Open Science Cloud is very, very helpful to see the European perspective, to see how data needs to be exchanged between countries, between people in Europe. And finally, I would like to thank here locally the Stephans and Uwe and his group for this very fruitful environment where I can develop these things, and also our funders. By that, I would like to thank you for your attention. And yeah, please ask any questions. I'm open now for questions. Thank you.
Thank you very much for your talk. So we got the YouTube stream running, but we do not have any questions on YouTube. So I say goodbye to our spectators on YouTube. Our next talk is on 16 March. It's about an introduction to knowledge-driven fair data services and the Leibniz Data Manager. So thank you for watching and goodbye on YouTube.